Okay, so a couple of things just to give you a quick couple of updates. First one is that folks have been asking for practice exams. There are 2 up now, so they're in the Unit 1 section, all the way at the end, below all of what we're doing, the review sections. There are 2 practice exams. They are from last year, so fall and spring. Be forewarned, we've not gone through everything yet. This is the last section of stuff, the microbial metabolism. So that's the first thing. The second thing with that is that those exams do not have dichotomous keys on them, because that is something that I used to cover much later in the semester. I took a look at when they appeared on exams, what the format looked like, and it's very similar to what we did in class on Friday. So use those and the extra ones I gave you as practice for that, and then, but that should suffice actually.
Do most people finish in the 50 minutes?
Great question, yes. And we'll talk about this some more later. But what?
They seem really long.
They're challenging. We'll talk more about this later. I will make sure to set some time aside on Friday for this. But what I do is I will show up in here at 12:50, and as soon as you come in, you actually can start taking the exam. So it's designed to take place in 50 minutes, and I take every single exam on my own beforehand, and if it's longer than 16 minutes for me to complete it from start to finish, then it's too long. Because I multiply my time by 3. Look, that's a rule of thumb I learned from one of my professors back in college. So like if it takes me more than 16 minutes to do, it's going to be too long. But I assume that it's going to take 3 times the amount that it takes me to do. But I am providing you from 10 minutes before class till I will stay in here till 2 o'clock. So you actually can have 70 minutes to do the exam. But we'll talk more about that on Friday. Nope. And guess what? Nope. Well, it can be blank, yeah. Blank sheet of paper. I know. But spelling doesn't count. Like, I'm not one that... Meaning. Come on, doesn't A&P force you to spell exactly correct? You guys should be thankful for that. Do you really want to spell allosteric and... Alright. It's just one less thing to worry about. Alright. Let's start with this. Well, actually, questions that you have on anything from that assignment. Parts of an enzyme, the different types of enzymes. Dan,
I'm confused between coenzyme and cofactor.
Yeah, great place to start. So coenzyme versus cofactor. Cofactor is kind of the broad category here. Coenzyme is a subset of cofactors. Okay? So both of them have the common function that they are required for enzymes to function properly, but they themselves are non-protein. So examples of cofactors can be, oftentimes we consider metal ions like magnesium, Fe3 plus or Fe2 plus, so iron, or zinc as 3 common cofactors. And I'll come back to the purpose of them in a minute. Coenzymes would include NAD, nicotinamide adenine diphosphate, and then FAD, which we talk about in the context of redox reactions or oxidative reduction reactions in biology. So the purpose of these coenzymes and cofactors, there are a couple of common roles. One is they are oftentimes charged so that they can be acceptors of electrons and also donate electrons back out. So, for example, we'll talk about later today the dehydrogenases, which are enzymes involved in redox reactions. They will have in their active site NADH or FAD that comes in, will accept electrons from something else, and then will move out afterwards. And the NAD, for example, go to the electron transport chain eventually. In the case of like zinc, magnesium, Those serve a couple of purposes. They can be acceptors and donators of electrons. We'll see that with the electron transport chain on Wednesday. They can just be there to stabilize the substrate in a certain conformation or orientation. And then sometimes they themselves are actually involved in the reaction beyond one of those two mechanisms. But the basic thing to know about them is they're required for the enzyme's full activity. Okay, and cofactors are, we consider cofactors to be organic and inorganic. Coenzymes, the subset, are just your organic cofactors. The ones that are made out of carbon, hydrogen, oxygen, nitrogen. Okay. Does that help, Daniel? Okay. Other questions? Okay. I just wanted to review a couple of pieces here. This is one of the images from the slides showing a particular enzyme. This one's got what's called the apoenzyme and the holoenzyme. Basically, the difference between the two of them is the apoenzyme is just the protein. The holoenzyme includes the cofactors and coenzymes. You really don't need to understand the difference between those. But if you were confused, that's what it means. This particular image here is showing those cofactors and coenzymes as two separate things. Again, the cofactors are typically ions. Coenzymes are typically molecules, NAD and FAD as the major ones. And then we've got a couple of sites. So this location is known as what? That's our active site. What binds there? Substrate, and what kind of inhibitor? Competitive inhibitors. Typically, antibiotics are competitive inhibitors of enzymes. And that's just something we'll come back to later on. But typically, antibiotics look like the substrate for some enzyme that is being inhibited. What is this site over here? That's an allosteric site. So what does that actually mean? What is, for those of you that defined allosteric site, what is it? So we can define it as where the non-competitive inhibitor binds. That's definitely part of it. That's not the full explanation. Other ones or additions?
I was just going to say that it's where the molecule that's not a substrate can bind.
That's a more broad definition, and I would tend to go towards that one and then supplement with the where a non-competitive inhibitor binds. And I say this because allosteric sites, it's not really easy to, on a protein, just point at something and say, yep, that's an allosteric site. An allosteric site actually gets defined by molecules that bind to the enzyme or the protein. So we often don't identify them until after we know what binds to that enzyme. Any site other than the active site, allosteric sites will change the activity of the enzyme. So what do we mean by that? A non-competitive inhibitor would bind to an allosteric site, and how would it change the activity? Would it increase or decrease the activity? Decrease. Inhibitors decrease activity. There are also allosteric activators. Molecules that when they bind to the substrate will increase the activity of, when they bind to the allosteric site will actually increase or enhance the activity of the enzyme. The way that I typically will think of this is from a couple of different models. I'm just going to draw a very crappy picture. So let's say this is an enzyme. Let's say this here is a substrate. Okay. And typically the substrate would enter into the active site of the enzyme, undergo some sort of chemical change. What a non-competitive or allosteric inhibitor does is when it binds to the enzyme is it actually changes or distorts that active site in some way. In this model here, all I'm trying to show is when you've got that, this red one is that non-competitive inhibitor or allosteric inhibitor. When it binds, it leads to closing of that active site. So no longer is it accessible to the substrate. And this obviously will slow down the activity of the enzyme overall. Activators, on the other hand, work in the exact opposite manner. Typically, the protein will adapt some sort of closed conformation. And when the activator binds to it, that protein opens up, allowing now for the substrate to come in. A classical example of this, if you've been in one, I can't ever remember if it's 151, 152, or 155, or any of the above. Do you guys talk about cyclins and cyclin-dependent kinases and cell cycle? No? Damn. Okay. Anyway, classical example of proteins always being expressed, but only turned on when an activator is present. Okay? And these activators can be other proteins. They can be other molecules. So sometimes it's a protein-protein interaction that leads to these changes. Let's take a look at an example of this real quick. There's one step within glycolysis where this occurs. Third step. Again, I'm not asking you to memorize these particular enzymes by any means, but this is a really classical example of allosteric regulation. So we've got phosphofructokinase, what kind of reaction does this catalyze? What does a kinase do? Transfers phosphates. Okay, transfers phosphates from one substrate. Typically ATP is involved in some way, so it might transfer it from ATP to, in this case, fructose 6-phosphate to form fructose 1,6-bisphosphate and ADP. And we'll see the inverse as well. In this case here, phosphofructokinase is actually regulated by 3 different molecules: ATP, AMP, and citrate. This is early in the glycolytic pathway. So this is highly regulated because this is in some ways you can kind of think of it like a rheostat, or like if you're turning up the temperature in your house because it gets cold, or turning down because it gets too hot. Phosphofructokinase does the same thing, but it senses how much energy does the cell need at any given time. How do we typically measure, what is our currency typically for energy in a cell? ATP, right? If ATP levels are high within the cell, if there's more than the cell is needing at a particular time, would you expect the cell to continue to produce more ATP through glycolysis? Probably not. We'd expect it to start slowing down those pathways. And that's exactly what happens. So when the energy level within a cell is high, when there's more ATP than that there's needed at any given time, ATP will bind to phosphofructokinase, distort the active site in a way so that it no longer can bind to the substrate, and therefore slows down these initial steps of glycolysis. Instead, this fructose 6-phosphate will get stored and then used in periods of time when that energy is needed. Okay, in our cells, it's stored as glycogen typically. Citrate is another inhibitor. Where does citrate show up? Not sure if anybody will know this one. Krebs cycle is also known as what? It goes under 2 other names. Citric acid cycle, right? Citric acid is citrate. It's one of the first molecules that's produced in that pathway. And so if there's a lot of citrate around, that tells us that there was probably an abundance of pyruvate as well, which is the last molecule produced in glycolysis. Therefore, there's a lot of energy eventually coming through respiration. And so again, to modulate how quickly glycolysis is proceeding, citrate will slow down those upstream steps to tell the cell, conserve your energy. Don't waste the ATP here to continue this process. The last one is AMP. Anybody know what AMP stands for? or what it is in relation to ATP? Yeah, ATP is adenosine triphosphate, AMP is monophosphate. So typically, this is kind of the relationship between the three. As phosphates are being taken off of ATP or AMP to perform some sort of function in the cell, eventually it gets converted to AMP. AMP is a signal for low energy conditions. And so the cell needs to resupplement itself with more ATP. In that case, AMP is an allosteric activator. It speeds up these reactions, thereby allowing for more energy to be produced in a smaller window or period of time. So we see these positive feedbacks, like in the case of AMP, that increase enzyme function, and negative feedbacks where enzyme function is slowed down as a way to constantly monitor and keep in check how much energy is being produced by the cell. And this is just one example. This occurs in many different metabolic pathways, but this is one kind of classical example. 3 major enzymes. I'm changing thought processes now. 3 major enzymes that I want you to know: kinase, isomerase, dehydrogenase. Kinases we've just kind of covered. They are involved in transferring phosphates. So what are you going to be looking for then if you think that a kinase is involved? ATP is going to be a prime, either being broken down to form ADP or being created. Both cases, we oftentimes say, yep, kinase involved in that process. How about an isomerase? What do isomerases do? Or what is an isomer? Okay, so isomerases basically break bonds and create a new bond within a molecule at a new location, right? What's true about isomers? If we're talking chemical isomers, think back to chemistry. Ugh. What is an isomer? Same what? Not same structure. Same molecular formula, different structure. Okay, so isomers have the same molecular formula, different structure. So isomerases, by breaking those bonds and creating new ones, change the structure but yet keep the same amount of, and what we'll see, carbons, hydrogens, nitrogens, oxygens, phosphate groups. And then the last are our dehydrogenases. What does a dehydrogenase do? Okay, reduces the electron acceptor. In other words, they are involved in what types of reactions? Redox reactions. I want to point out here with dehydrogenases, also in the name is dehydrogen. If you see in biological redox reactions, if electrons move, hydrogen is moving as well. So if, for example, we see something like NAD going to NADH. Oh, shoot. Oh, damn. What's not drawn in this reaction is that we have to have a hydrogen and 2 electrons being transferred from another molecule to produce the NADH. So NADH and FAD, or NAD and FAD, are going to be found as coenzymes within dehydrogenases and are also involved in redox reactions. So the reason why I'm doing this is because one of the things that I'm going to expect that you're able to do is not to necessarily just say what the enzyme does. I'm not asking you again for specific names of enzymes. Instead, what I'm asking you to be able to do is to go to a picture of glycolysis, like the one you got in front of you, and identify these different types of reactions. So in which ones are there kinases being involved? In which ones might a dehydrogenase be found? And you'll see examples of this on the practice exam too, where I've either left out the enzyme name or left out one of the substrates, and you've got to fill in the blank given these particular word associations. So that's what we're going to practice with for the next 15 minutes or so. We'll go till just after 11 or 1:40. This worksheet is kind of designed to get you to think about these connections between kinases, isomerases, dehydrogenases, yada yada yada. Work alone, work with a partner, I don't care. But work on it for a while, and then just so that you are aware, at the end of this section, the microbial metabolism lesson 1, there's the key to this. Don't look at it now. It's not going to serve you any good just to do that. But there's also like a 10-minute video where I walk through the answers to this if you need more explanation than just what's on the key itself. Okay? All right. We'll take 15 minutes and then discuss. How do you tell? We'll go over it. We'll go over it. Don't freak out yet. Or if you are, just do it silently. Okay, there we go. All right, so let's take a look at this real quick. What I want to start with is let's kind of break down the 3 major types of enzymes again and identify where they're involved in glycolysis. So, kinases. Where do we find kinases involved in glycolysis? Where there's phosphate or where there's ATP. So tell me, which steps are they occurring in? First one, the enzyme there, not that you need to know it, hexokinase. Nope. All right, so 1, hexokinase. What else? 3, phosphofructokinase. 7, yep. Even when we get ATP out, there's a kinase involved there as well. 1 more. 10, perfect. Okay, so 4 different kinases are involved in glycolysis. These drive what we call substrate level phosphorylation, okay, as opposed to oxidative phosphorylation, which we'll talk about on Wednesday. In substrate level phosphorylation, all we see is a phosphate being transferred from one substrate to another to form 2 new products. So if you're trying to do some more word association, substrate level phosphorylation, you will always see a kinase involved. Okay. How many ATP are we putting into this system? 2. We call this first stage here. Typically it's kind of the energy consuming step. And then energy output step. How many ATP do we get? 4. So net we make 2. All right. I think that's all I wanted to cover with that. How about isomerizations? Where do we find isomerases being involved? 5 is one of those steps. Yep, this right here. Which other one? Yep, right here too. These ones, how do you identify an isomerase reaction? The easiest way to do it is really just to count up everything that's there. Count up the carbons, hydrogens, and oxygens, and of course the phosphate groups in these as well. And make sure that they're the same number of each. And if there is, then an isomerase has been involved. Sometimes the names of the molecules will change, will tell you as well. So for example, in Krebs cycle, we see citrate and isocitrate. They're isomers of each other. And so there's an isomerase involved in that reaction too. All right, dehydrogenases. What are we looking for? We're looking for NAD, NADH, so where do we find that? Oh, yeah, number 6, right here. So basically in this reaction, we have NAD, which we defined earlier as a coenzyme. It's also going to be our electron acceptor here. We have glyceraldehyde 3-phosphate. It's going to donate its 2 electrons. It's going to donate 2 electrons and a hydrogen to NAD plus to produce NADH. Couple of mnemonics that people typically use to remember redox reactions. Leo says GER, loss of electrons is oxidation, gain of electrons is reduction, or oil rig. Oxidation is loss, reduction is gain. There is one other spot where redox reactions do occur. It's not technically in glycolysis, it's actually in fermentation. And this occurs when pyruvate undergoes a fermentation. So it will be reduced, thereby regenerating oxidized NAD to continue this process. And we'll cover that on Friday. One other thing I want to point out, because I'm realizing we haven't done this yet, but there are a couple of redox questions at the end here. I wanted to just point out a couple of things for you real quick. First, I'll just write down the mnemonic. Loss of electrons is oxidized. Gain of electrons is reduced. Okay. In a redox reaction, there's always going to be a product that's oxidized. There's always going to be a product that's reduced. So, reduced and oxidized. Also in these reactions, there will always be an oxidizing agent, which is one of the reactants, and there's always going to be a reducing agent. The reducing agent is what donates the electrons. And so the reducing agent, by losing its electrons, will become the oxidized product. The oxidizing agent, because it's going to gain those electrons, will become the reduced product. I bring this up because these redox reactions are central to Krebs cycle and more so to electron transport chain, as those high energy electrons are used to fuel the pumping of hydrogen ions to the inner membrane in between the inner and outer membrane on gram-negative bacteria. So take a look at those examples, take a try with them, and let me know if you have questions. The last thing to mention, I did post a quick video on this too, so if you don't pay attention right now, that's okay. No more voice threads until after the first exam. F voice threads, FVT. Okay.